Hey builders, in this Retro Reacts, we're going to be taking a look at another one of the early 2017 This Is My Architecture videos. In this one, we're looking at Station X's gene pool solution, where they're analyzing genomic data at scale, leveraging AWS. This should be very, very cool. Let's check it out. Just like I said.
Welcome to This Is My Architecture. I'm Matt, and today I'm joined by Anish from Station X. So tell us about Station X.
Yes.
So we got Matt back. We're going to hear from Anish and get that little blurb. I like this. They kind of orient you if you've never heard of the company before, so you kind of understand what vertical they're in, what industry they're tackling, like sort of what problem they're trying to solve.
Station X, we're building our platform called Gene Pool, and we're focused on building a platform for analyzing, visualizing, and managing genomic information.
Okay, so the cool thing is for genomic data, there's a massive amount of it. So when you're doing any sort of bioinformatics or biotechnology experimentation or analysis, you've got a massive amount of gene pairs that you need to coordinate, run a whole bunch of probabilities on. It's a really cool problem set. There's been a ton of great AWS case studies. You should check it out, and let's see what they've got in store for us today with this gene pool solution.
Very cool. So you built this genomic analysis platform on AWS. Tell us about the architecture.
Yeah, so I think probably the best place to think about is where is this data coming from? Yeah. So the data ultimately is patient samples that are sequenced on machines, and often
So patient samples that are sequenced on the machines, most of the time the output of these sequencers is literally going to be text files. And I know that doesn't seem like a big problem, except you realize that it's gigabytes of text that is just GACT, right? It's just the genome pair sequences, and you need to figure that out and line it up based on these different base pairs. And then depending on what type of sequencing they're doing, they're going to be looking at a different gene expression. So it's a relatively simple data type, but the volume and the ways that they have to analyze it make it into a really fascinating computer science problem.
Sequencers now stream the data directly onto S3.
Oh, very cool. So sequencers themselves actually support S3 as a target?
Exactly.
Oh.
That's super cool. Like the sequencers dumping directly at S3 saves a whole bunch of steps for the scientists, which may not have the IT support or the cloud knowledge to actually set this up themselves.
And that's a very raw form. And then we have a number of partners that we work with, such as DNA Nexus and Seven Bridges. And what they're focused on is assembling that data, mapping it to the genome, and finding positions of the genome that vary between the patient's genome and a reference genome.
So that's when they're looking at the model of like, okay, this is the standard based on the Genomics project, mapping out the human genome. This is what this patient is expressing differently. And then here's what we know about those pathways. So part of these services in the back end, what they're going to be doing is saying, if 1, 2, 3, 4 is expressed above this certain point, that's an indicator for this type of issue. There's, like I said, a huge amount of data assigned to this, which is really interesting.
I don't understand genomics. Are these essentially like text files stored in S3?
Yeah, they're essentially text files.
Really big ones.
Exactly.
Really big and highly compressible text files.
Okay, cool.
So at that point, we're ready to pick up that data in Gene Pool. In Gene Pool, we're focused on what's called tertiary analysis. That's actually like real interpretation of this genomic information.
Okay.
So at this point, what they've done is they've done the ingest of the data, then they've done a bunch of enrichment basically through their partners, and now they're getting to the actual analysis phase. Let's see how they do that.
So it's making sense of the raw data.
Exactly.
So you have the raw data on S3 coming from your partners or from the customers themselves. So what happens next?
Yeah, so at that point, someone could use our APIs, or they could log into Gene Pool, and they would select a set of patient samples that they're interested in. It could be on S3 or DNA Nexus and Seven Bridges.
Okay.
At that point, the data is ready to be imported in Gene Pool.
Okay.
And what we would do is we'd spin up our EC2 servers to process that data. So at this point, we're just doing some raw processing of the data, some validation, and some normalization of it.
Okay, so this first step, they're doing a bunch of data cleanup, essentially, which is, 95% of machine learning and big data projects, it's all cleanup. So they're doing that with this first set of EC2 instances.
On a pre-processing. What are you actually using to do the pre-processing?
Well, this is some of our own custom in-house built tools.
Okay, so you wrote your own code there.
Exactly.
So the pre-processed data, I imagine, then gets
Yep,
dumped out to another.
exactly. So at this point, it's still just text files that we have on S3.
Okay.
But text files aren't the most optimal thing for querying. And so one of the things we do is we use Hive as kind of our ETL process. So
Okay, so what they've done is they've done the ingest, they've done the cleanup, they've dumped it back into S3 still as text files, and then they're going to convert it to a more usable format, in this case Hive, which is pretty popular with some big data tools around this time. You don't see it as often right now, but it's still a highly effective big data format.
It's going to do some aggregation of the data as well as convert it to ORC.
So where are you actually running Hive?
Yeah, so we're running Hive on EC2 servers, and the way we do this is we're using Kubel to manage that. So they're responsible for spinning up our Hadoop clusters.
Cool, so Kubel is a managed big data service, you're using it to run Hive. Again,
Okay, so they're using this other tool, Kubel, in order to run all their Hadoop clusters to pull the data out of the Hive format or use Hadoop to access that Hive data to start doing the analysis, all that mapping and reducing to figure out what the answer for the actual query.
that you have in your
Very
S3
cool.
buckets.
That's right.
Got it. So what do you actually do with the data? What happens to it?
Yeah, so what we do is we will generate these ORC files.
Is that the new Office Space line? What do you actually do with this data? It's a good one. I'll keep that in mind.
And there's a couple things that we do that are kind of unique. So one thing we do is we'll partition the data by chromosome.
Oh, that's interesting.
And then the other thing we do is we'll sort the data by genomic coordinate. And this is really important because one of the things that ORC files have is a concept of stripes. And
So what they're doing here is they're basically using Hadoop to take that those files and line them up, which is a great practice in any data project, let alone a big data project, to line it up and in its storage with how it's most often queried. So for genomics researchers, you're going to be going by a genomic coordinate or by chromosome, most likely as your first point of search. So they're trying to store the data in anticipation of that, saying if we line everything up in these rows, it makes it easier for people to come down and figure out what they want, which is a really smart thing for optimizing the user experience.
And so by sorting it by coordinate, one of the things you can do is rather than have to scan all your data, you can actually just jump to a particular stripe that has the
jump
data
to
of
the
interest.
chromosome or the
There you go. So what they've done is they've optimized that data, not just for read, but also for storage. So they can make sure that instead of accessing everything, they're just accessing that one part of the data store system that has the contents that the user's interested in.
the chromosome.
Exactly. And so it makes it a lot more efficient.
I love that. That's super cool because normally I work with customers like log files and they're kind of partitioning by date and maybe the stripes or the sorting is done by alphabet, but this is for genomics. I love that sort of marrying of biology to Presto.
Yeah. So what Matt just called out there is spot on. If you've gone through any of these projects before, that general technique makes the most sense. Figure out how people are going to be querying it and store it that way because it's going to be read far more often than written for use cases like this. Though in a previous Retro Reacts, we did see the opposite. We saw a case where data was written far more than it was read, and so the design in that case optimized for the write. But most of the time, you should be optimizing for read.
Cool. So you have these ORC files, these compressed or optimized files for querying. What happens then?
Yeah, so at that point, what we do is we use
Side note, that's a fantastic question, not just for architecture, but for security as well. What happens then? What happens then? Just keep asking that until you end up at the end of your solution.
for our live, interactive, near real-time querying of that data.
Okay.
Near real time. Did you catch that? Near real time. Just given the volume of data, it's not going to be purely real time. They get it pretty close, I think, at this, but with tools you'd have at your disposal today, you could get it to real time.
Again, we're managing these Presto clusters through Kubel.
Okay.
The way our customers interact with it, our customers are scientists from biotechs, pharmas, and clinical diagnostic companies, is that they'll log into GenePool and they will select a set of samples they're interested in analyzing. And our GenePool analytics servers will communicate with the Presto master node to do the analysis. And
Okay, so they've written a custom front end in order to help have a multi-tenant system here so that they're maximizing their resources, but also to line up those requests with the area of the data store that has that data, which totally makes sense.
Scientists are not actually issuing SQL queries, like you're actually converting it to Presto on the back end?
That's right.
Got it.
So, a lot of our users are, bench scientists who maybe aren't, don't know how to do the SQL queries or don't know how to set up this big data infrastructure.
Yeah, they're just
Or they don't have time and don't care. That's the other thing I think in IT we generally need to figure out is like, hey, there are better ways to deal with this stuff rather than low level. So sure, writing a SQL query might be more efficient, but a Presto query is a little more understandable, especially if it's not your thing. If your data analysis isn't really your thing at that low level, if you're a much higher level and you have that subject matter expertise, you're like, I just want to ask for this, this, and this. You should be able to ask in a language like that. Presto's not bad. There's a bunch of others that I find are better, but it really depends on your audience, right? If your audience is familiar with it or can adapt to it easily, or it's close to something they like, use it. Go for it.
results. They want to visualize it.
Exactly.
Okay, cool. So you make it easier for them to really sort of
That's
visualize what
right.
they need to see, get to the point of the chromosome they need to get to.
That's right. There's, of course, the analytics tools, some of which is built on Presto, but we also have a very rich user interface that allows the user to visualize their data, allows them to interactively query and filter their data so that they can.
So this, I can't see all the details here. This is the one downside of this density of data, but this looks like sequencer output where you're running a bunch of plates and you're looking for what area in what well is reacting to the different filters that you're putting in place. But this gives you just an inkling of how dense the data is, even though it's all originally just text files. It's just a crazy amount of density here.
Come up with a set of variants that are interesting, maybe
like the graph.
causing a disease, or look at differentially expressed genes to build by.
So this is way more useful for a scientist or a researcher than writing SQL queries, right? Having that rich visualization, I can see why this was a popular solution.
Markers.
Very cool. So are most people interacting with the UI and then a press on the back end, or do you have sort of an API layer?
Yeah, we have both. So we have customers who are using the UI, but then we also have some customers who are using the APIs, and we have integration with Deloitte Converge Health's TRIP ecosystem, which is focused around patient data, but then from TRIP, you can jump into GenePool to look at the actual genomic data.
Oh, so that's interesting. So not only do you integrate with partners on the front end for their raw data, but you integrate with third parties as well on the final end after the processing.
Exactly, that's right.
That's very interesting. Well, I love how you're using Qubole with Presto and Hive. I especially love how you're doing the partitioning and the striping, mapping it to genomics. This is a very interesting architecture. I really appreciate you taking the time.
Yeah, that was really, really cool. I like that. I like when we're outside of IT and we're looking at different solutions and different challenges. In this case, we're looking at genomics data, which while it seems really simple because you're like, oh, it's just a bunch of text files with some letters, the density and the volume is crazy. So having that ingest or all those actual sequencers are dumping into S3, then they're doing the cleanup with EC2, putting it back into S3, then pulling it into Hadoop clusters through the Hive format and allowing Presto to coordinate some stuff to make it easier. And then they have that nice slick UI for the analysis. Very, very cool solution for the day. Again, this is 2017. What would I do different? Pretty much everything. I would still have the sequencers now today dumping into S3, but I wouldn't do the custom cleanup in EC2. I'd be using Data Glue and Data Glue Brew. Glue data. There's a bunch of rhyming there, but AWS Glue and then Glue Data Brew helps you do the cleanups in a visual manner and then automate it after the fact. Those tools have matured. They weren't even in existence in 2017. They are far mature now. They're great, slick provisioning there. The question I would have then is, even would I have to get to the point where I'd have custom visualization? This may be pushing a service like QuickSight to its limits. Or even beyond its limits. But I'm thinking what would be a really strong solution here is you're basically getting to the data lake and to the lake formation idea where you've got all the sequencers pushing into S3, doing cleanup in AWS Glue Data Brew, and then in S3 you can use Athena to chunk it up into different areas. Now you may again have to do that conversion away from text files into a different format to sort of optimize, but you could use Athena to do that same sort of striping between chromosomes or genomic coordinates, and then you may have to slap a visualization front end on it, or you may be able to push QuickSight to its edge in order to get that type of analysis, but you'd probably still have to have some sort of custom UI in front because the volume and the density of the data is not what QuickSight was necessarily designed to handle, but it'd be worth a shot because if you could get it there, your costs would be significantly lower. But if you did have to go to the custom front end, I'd containerize it all. If you couldn't do serverless, you'd try for serverless, but again, you may be hitting, as far as the volume of data and the wait times, you may be pushing it kind of to stay in Lambdas as your compute engine, but having the containers there would actually reduce your costs. You get some efficiencies in place. So you could rip and replace a lot of this. You could get away from the Hadoop, get away from Presto, and make this a lot simpler. So it's easier to run. It better aligns with the AWS Well-Architected Framework, but the principles will still be the same. Ingest, cleanup, optimize, and then allow that analysis and manipulation. But I love these kind of solutions, super, super cool. If you're interested in genomics on AWS, there's a ton of great use cases there. I'd also highly recommend checking out the work by fellow AWS hero Ling Langit. She has done a ton of great consulting work and written a lot of great stuff around how to do genomics and big data at scale in AWS. I'll put those links down in the description. And with that, another great architecture that we've taken a look at in this Retro Reacts. I'll see you in the next one.